We will continue our discussion about this Calnexin role in the proper folding of these GPI-APs. In this video, we will discuss within this proper folding whether the Calnexin helps these GPI-APs in deacylations or not, and how this Calnexin will help. Calnexin will help using Glycan interactions that are expressed on the GPI-APs when they are translated on the endoplasmic reticulum. So how they did their study is that they identify that Calnexin, they identify or understand from the literature that Calnexin has different domains. First one is glycan binding domain or globular domain that interacts with the glycan structure, that is glucose 1, mannose 9 and lignac 2 structures and glycan structure that are expressed on the amino acid translated in the endoplasmic reticulum of these GPI-APs or other glycoproteins. The other things that uh, other domain that Calnexin has is one of them is P domain that helps in properly folding the disulfide bonds, and so our focus will be on these two domains of the Calnexin for this experiments. So first they took this globular binding domain where they have identified from the literature that what are the amino acids that plays the roles towards the glycan interactions, and they have find out this Y164 and K166 and M188 and E216. These are the four amino acid that helps in the glycan interactions to the Calnexin. So they need to check whether GPI-APs can properly deacylated without the Calnexin interactions or not. So in order to do that, they prepared a Calnexin that has the mutations in each of this site with the annealing that is shown here in the panel A, and then they check by expressing this defective Calnexin in a Calnexin and calreticulum dual knockout, and they check this remaining uh, GPI-APs expressed on the cell surface after PI-PLC treatment. And in order and what does this PI-PLC does is the PI-PLC can clip off this mature GPI-APs or properly folded GPI-APs. However, immature GPI-APs that are expressed on the cell surface will not be cleaved by this PI-PLC. So what they have find out is that in all the four mutations, they see that the clipped off of this GPI-APs are, or in this case, the CD59 they checked. The clipped off of this CD59 is negligible or is almost no clipped up occurs compared to the wild type, or we can say that the remaining CD59 is statistically significant compared to the wild type in all the four knockouts. And so after validating that the interaction of the glycan, the interaction of this Calnexin with the glycans of the GPI-APs is necessary for proper folding or deacylations. And after that, they need to check whether this disulfide bond interactions, which is usually been done by ERP 57 or ERP 57, yeah, ERP 57 interacts with this Calnexin, and thereafter that complex can interact with the glycoproteins that helps the properly disulfide bond formations. So they also identified what are the amino acid or the P domains plays a role towards this ERP57 interactions to form a complex, and then that complex can help the proper disulfide bond formations of these glycoproteins. So they identify those lectins or not those amino acids that plays a role towards this ERP57 or disulfide bond interactions. And what they find out is that some of these construct are not stable, however some of them are stable, and then they check what are the constructs that are stable, and they can see that in the panel B shows which are the construct that are stable. So they express this construct in the Calnexin and calreticulum dual knockout. So this E351A and this D343A, and this is the dual knockout D343A and E351A, and are stable. However, very other ones are less stable, as we can see that D343A single mutant does not have any calnexin even after it is expressed in the X cells. So after identifying which are the construct that are stable, they check the same thing, that is CD59, remaining of the CD59 after this PI-PLC treatment. And for the wild type cells, they can see that the PI-PLC are been cleaved off almost to all extents. And even in the dual knockout where calnexin is uh, transfected as a wild type, they can see that there is a cleaved off, or this remaining of this CD59 is very minimum. However, for some other case, in this D343A, since the calnexin is not absorbed, they can see that it is also cleaved off to some extent. However, for some constructs such as W342A, D343A dual knockout, they can see it's partially recovered. This the CD59 are been partially recovered. Remaining of CD59 after PI-PLC treatment is not completely goes down to the background signal. There is a partial misfolded CD59 are expressed on the cell surface for these dual knockouts, and also for the single knockouts, we can say that from these results. So after understanding that the disulfide domain, after understanding that it's the disulfide domain might be helping in the, after these results there are two kind of hypotheses that can be formed. First one is that this disulfide bond helps in this proper folding of these GPI-APs or deacylations, or it disrupt this proper folding of GPI-APs or deacylation. So they work on the second hypothesis where they knocked out this PDI-A1. They knocked out this PDI-A3 gene that is responsible for producing ERP57, and that interacts with the calnexin, and thereafter that complex helps in this proper disulfide bond formation. So they need to check the second hypothesis, and they can see that in the wild type case there is a uh, there is a recovery of, or the signal of the CD59 after PI-PLC treatment goes down, and this in line with this PDI hex cells where PDI-A3 has been knocked out. This indicates that the second hypothesis seems to be true, that ER57 disrupt, or ER57 is not required for the deacylation of these GPI-APs. And same things they have validated using CD55 also, where the signal goes down and it is in line with the wild type signals. And these things are been quantified in this panel E, where we can see that the signals even goes down below the wild type. So this ERP57 is causing disruptions in these deacylations formations. So that's why this in the case of this PDI-A3 knockout, the signal goes down even below the wild type. Then the signals of this remaining of the CD59 after PI-PLC treatment. Thereafter, from the literature, they need to check whether this PDI-A3 also interacts with this soluble misfolded protein, or they only interact with the misfolded proteins. So in the immunoprecipitant assay, they first so they created two different kind of cells. First, two different kind of CD59, two different kind of CD59. First one is this C94S, which is misfolded protein, and second one is this C94S and G103 stop codon. So this is the soluble misfolded protein. So in this case, they identify that in the case of this misfolded CD59, they can see the interaction of the calnexin with during the immunoprecipitant of the flag. They can see the interaction of the calnexin, this interaction during the immunoprecipitant of the CD59. Yeah, that's right now I'm so correcting it. So during the immunoprecipitation of the CD59, they can see the calnexin presence whenever there is a misfolded CD59 is there, and when the misfolded and soluble CD59 is there, there is no calnexin interaction occurs. And same things they have identified for these PDI-A3 knockout cells. So this thereby indicates that calnexin interact with this Glycan structures or misfolded Glycan structures is not dependent on these disulfide bond lactanes such as ERP57 that helps in the uh, properly folded disulfide bonds. So this panel F further substantiate their results from the panel D and E that ERP57 is not essential or not necessary for the deacylation of these GPI-APs. Um, so, however, they did not explain the reasons why this soluble CD15 does not interact with the calnexin, even in the case of wild type and even in the case of this PDI-A3. One of the reasons could be that because of being soluble, they might be directed towards the ERET pathway, but they did not mention so far in the paper. We will discuss furthermore uh, about this topic in our next videos. However, if you want to understand the experimental procedures and the results in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.